we talk about inheritance, we have to define some words first to make sure that all of us are on the same page. The first word I want us to talk about is genotype. So a genotype is composed of all the chromosomes and all of the genes that are contained on those chromosomes. And a genotype is unique. It's unique to an individual. So it determines your anatomical, physiological characteristics. And to refer back to what I've talked about before, no single cell or tissue uses all the information contained in a genotype, because we have differentiation and different cells do different things so that different tissues do have different functions. A person's genotype is derived from the genotype of the parents. You essentially got half your DNA from your mom and half from your dad. So this karyotype that you're looking at here is all of the genetic contents, all of the DNA in one nucleus of one cell. This DNA came from a male. As you can see, we have the XY is indicated right here. Notice how these chromosomes are lined up. They're lined up from largest to smallest. The centromeres are directed more toward the top, if you can tell that. So they're by size and centromere placement. The phenotype of an individual is what you see, or the physical expression of the genotype. So it's the anatomical and physiological characteristics. So to refresh you just a little bit, remember that your DNA would be indicated by this double strand piece of DNA that when you take it apart, you're looking at the building block, which is a nucleotide. Each single strand of DNA is made by connected nucleotides connected at the sugar phosphate backbone that is indicated here. The double strand is two single strands that are joined together at the bases. Notice this is a C and G, this is a T and an A. So all of your double-stranded DNA is found in the nucleus, and when it is most condensed, you see it as a chromosome, right? So this is when you would find it most condensed during metaphase of mitosis. And all of your DNA is found within the nucleus of a cell. So the lighter blue indicates the nucleus and the darker blue indicates the cell. Each one of your chromosomes is this is a replicated chromosome, is made up of two chromatids. So we have chromatid 1 and chromatid 2. Chromatid 1 is perhaps indicated over here, all right? So with this panel, I want you to see what an allele is. So this is the location of flower color here and the location of flower color here. So the genes are the same. The genes are for flower color, but the allele is an alternate version of a gene. So this is a gene for a purple flower, and this is the gene for a white flower, okay? So the term homologous chromosomes means the member of each pair of chromosomes. So this is homologous pair 1, homologous pair 2, homologous pair 3, etc. So in every single somatic cell, you should have 23 pairs of homologous chromosomes. That's 22 autosomes, right? So that's 1 to 22. Those are the autosomes. And then we have pair 23, which are the sex chromosomes, right? So autosomal chromosomes, they affect somatic characteristics for the most part. And each chromosome in the pair has the same structure and they carry the same genes. But remember, the alleles can be different. The sex chromosomes are the last pair of chromosomes that you would see in a karyotype, and it determines whether an individual is genetically male or genetically female. So here I'm showing you two different karyotypes. One, the dark one, the black one, is an actual karyotype of actual chromosomes, and the blue and beige is a cartoon rendition of what a karyotype would look like so that you could see it a little more clearly. So each one of these is a representation of a karyotype. Remember, I told you that they're numbered by size and centromere position. And you can see here the 22 autosomes above this line, and then here the sex chromosomes. Also, when we refer to chromosomes within a karyotype, I've already mentioned genes and alleles. Well, we can say a locus is the gene's position on a chromosome. If I wanted to say that the gene for eye color is located on this fourth pair of chromosomes, I would tell you that the locus is here, 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 and here. So for each gene, you have 4 copies in a somatic cell. Alleles, however, are alternate forms of the gene that affect the gene on the phenotype, that determine the effect of the gene on the phenotype. So here's another image where you can see that we have homologous chromosomes, but these are unreplicated. And so in this image over here, you actually see them as replicated. So let's say that this is the gene for eye color. So on this chromosome, you have 2 dominant alleles, a big G and a big G. And over here, you have 2 recessive alleles, little g and little g. Let's say that this is the gene for freckles. So big R means that for this allele, they're both dominant and they're both recessive over here. So these chromosomes would be part of a homologous pair. They have been replicated. So if we were going to talk about the genotype, we would say this pair gives 1 big G, because there's no reason to write it twice, and 1 little g. So this would be the genotype for this characteristic. I do believe I said it was eye color. I hope I did. So if it is eye color, then the dominant eye color is brown. So this individual would actually have brown eyes because they're big G, little g. Now, if they were big G with 2 G's, 2 big G's over here, they'd have brown eyes. And if there were 2 little G's over here, they would have blue eyes. Remember, you have 2 copies of each allele because we have replicated chromosomes that are part of a homologous pair. Homozygous means that both homologous chromosomes carry the same allele. So if you look down at this picture, this is homozygous, right? But if we're heterozygous, both homologous chromosomes carry different alleles. So in each one of these images, what you're seeing is unreplicated chromosomes. So I could put another chromatid here and another chromatid here, and you would have another big B over here and another big B over here. So you're just seeing unreplicated chromosomes. Could do the same right here and put a big B over here. Same right here and put a little b over here. All right, so these individuals that I'm circling right now are considered heterozygous. If you go back to the last slide, when you look at the G's, that individual would be considered heterozygous. We can put the extra chromosomes on here, and so we'd have another little b and another little b. Just remember, all the blue ones indicate unreplicated, and what I did by drawing those chromatids on there was to make them replicated. So simple inheritance means that the phenotype is determined by interactions between a single pair of alleles. So here's a really good example. A person either has freckles or they don't. That is single gene or simple inheritance, right? You don't have the in between. Hair color is not simple inheritance. It is polygenic inheritance. You don't just have blonde hair or black hair, right? You have all the colors in between. How tall a person is determined by many genes. There are certain things that are determined by single gene or simple inheritance. Whether you can curl your tongue, what color your eyes are. Let me just go to that for just a minute. Your eyes, y'all, are either blue or green. If they're brown, they're green. Sorry, let me say that one more time. Your eyes are either blue or brown. Brown is always dominant. If your eyes are green, then they're actually brown. If your eyes are gray, then they're actually blue. All right, so there's really only two colors of eyes, and that is single gene inheritance. Hair texture is polygenic inheritance. All right. So if you have a situation where you have strict dominance, that means the dominant allele is expressed in the phenotype regardless of the information that's carried by the other allele, such as blue eyes and brown eyes. That the brown eye is dominant and that what you're going to see is the dominant eye color, all right? Recessive alleles are expressed only if they're present on both chromosomes of a homologous pair. So if you look at this image, again, we're looking at missing chromatids, right? Same for this one over here. So you would then be looking at a homologous pair. So when we look at this gene, whatever it's the gene for, it's homozygous dominant. This is homozygous recessive. This one's heterozygous. In this image, this is the chromosome that came from the dad. This one over here is the chromosome that came from the mom. So if we're dealing with different forms of dominance, we can have incomplete dominance. So with incomplete dominance, you get heterozygous alleles that produce a unique phenotype. All right, so incomplete means that if you cross a red flower with a white flower, you get a pink flower. So ink pink. Codominance means you get both the recessive and the dominant phenotypes expressed. So if we use a Punnett square in order to predict phenotypic characteristics, if mom is recessive, we put her up here. That's her genotype. Dad is homozygous dominant. We put him down the side. You bring him over here, her down here, same thing, him over here, her down here. Do this for every box, and you will see that you have a 100% chance that the child born from a fertilization event between these two people would both be heterozygous. So if we now assign a characteristic to the different alleles, we're looking at the gene for albinism, and albinism is a recessive trait. So if dad is a heterozygote, he has a big A and a little a, and mom has albinism, she has a little a and a little a. You do the cross again, dad come here, mom down here, dad over here, mom down here, dad here, mom here, dad over here, mom here. You'll see that with each fertilization event, there is a 50% chance that the child will be born with albinism. So looking at the major forms of inheritance, if we look at simple inheritance, single gene traits, we have all of these down here at the bottom, these different characteristics that will be produced. So, for example, if we're looking at simple dominance recessive inheritance, freckles is dominant. Nearsightedness is dominant. Curly hair is dominant. Straight hair is recessive. It's actually dominant to be nearsighted. Neither one of my parents were nearsighted, but both my brother and I are nearsighted. So that indicates to us that our parents were heterozygotes for nearsightedness. Codominance, Your structure of albumins and transferrins and AB blood type. Remember, you can be A or AB or B or O. That is a codominance-dominant situation. In complete dominance, we have two different alleles. They produce intermediate traits. Examples are hemoglobin A and hemoglobin S. So when we're looking at polygenic inheritance, again, hair color indicates that it's many genes that cause this. For sex-linked chromosomes, as you well know, red-green color blindness, some forms of hemophilia, and some forms of muscular dystrophy. So again, polygenic inheritance, it's several genes. You can't predict it with a Punnett square. During meiosis, something occurs, it's called genetic recombination, which is where chromosomes change, they swap portions, and it increases variation, which is great, but it also complicates the inheritance of genetic disorders. So if the blue chromosome indicates the genetic information that came from dad, and the red indicates what came from mom, during meiosis, something called recombination occurs where a portion of each one is swapped. So that's what you're seeing in this image. So parts of chromosomes become rearranged during synapses. Sometimes chromosomes don't act the way that would keep a person in homeostasis. So you can damage chromosomes, you can break chromosomes, you can have pieces that are missing, and you can have extra copies. And these chromosomal abnormalities make it difficult for a fetus to survive. And different clinical conditions can result. So anything that you do, whatever the chromosomal abnormality is, you're changing the nucleotide sequence of an allele, all right? Sometimes mutations are spontaneous. They're not passed down through a family. Another term for these mutations is called de novo mutations. So these are random errors. They just occur in a population. X-linked genes, I just referred to them earlier. So these are the sex chromosomes, and inheritance doesn't follow the same way that it does for autosomal chromosomes. So if you look at this Punnett square, we have a female who is heterozygous, she has big C here, little c here, and a male that just has a big C, right? So when you make the cross, you get a normal female, and you get a normal female that is a carrier for whatever the disorder is. Then you get a normal male, but then you get a colorblind male who is going to have the disorder. He does not have the opportunity to have a big C to offset the effect of the little c. So this female here would be considered a carrier. She's heterozygous for the abnormal allele, but she doesn't have the mutation.